Hello, everyone. Welcome back to Meds.Concepts. Today we are going to discuss an extremely important topic in molecular biology. Replication in eukaryotes. Replication is the process by which a cell copies its DNA before cell division. In eukaryotes, this process is highly organized and tightly regulated because eukaryotic genomes are very large and chromosomes are linear. Unlike prokaryotes, eukaryotes possess multiple chromosomes, multiple origins of replication, nucleosomes, and complex chromatin organization. Therefore, replication in eukaryotes requires many specialized proteins and regulatory mechanisms. Let's see the overview of eukaryotic DNA replication. Eukaryotic DNA replication follows the semi-conservative model. This means each daughter DNA molecule contains one parental strand and one newly synthesized strand. Replication is bidirectional and starts simultaneously from multiple origins on each chromosome. Because eukaryotic chromosomes are extremely large, replication from a single origin would take too much time. Therefore, multiple replicons are used. Replication occurs during the S phase of the cell cycle. The process is highly regulated because each origin must fire only once per cell cycle. Replication speed in eukaryotes is approximately 100 nucleotides per second, which is much slower than bacterial replication. However, the presence of multiple origins compensates for the slower rate. Origins of replication and ARS. In yeast, replication origins are called ARS, autonomously replicating sequences. These are specific DNA sequences capable of initiating DNA replication. The ARS contains several domains: domain B1, B2, B3, and C domain. The domain contains the essential ARS consensus sequence where proteins bind. The most important protein involved is the origin recognition complex or ORC. ORC remains associated with replication origins throughout the cell cycle and acts as a platform for assembling initiation proteins. Each replication origin functions as an independent replicon. Very important difference from prokaryotes is that eukaryotic replication origins are much more complex and less sequence-specific. Initiation of replication. Replication initiation occurs through highly coordinated steps. First, ORC binds the origin DNA. Second, CDC6 and CDT1 proteins are recruited. These proteins help load the MCM2/7 helicase complex onto DNA. This assembly forms the pre-replication complex or pre-RC. Pre-RC formation occurs during G1 phase. During S phase, cyclin-dependent kinase or CDK and DDK kinase activate the helicase. Additional proteins including CDC45 and GINs associate with MCM to form the active CMG helicase complex. A critical concept is replication licensing. Each origin fires only once per cell cycle to prevent re-replication and genome instability. MCM helicase and replication fork. The MCM2/7 complex is the major helicase of eukaryotic replication. It forms hexameric ATP-dependent motor protein complex. Its function is to unwind double-stranded DNA ahead of the replication fork. As DNA unwinds, single-stranded DNA templates become exposed. Single-stranded DNA is unstable and prone to degradation or secondary structure formation. Therefore, replication protein or RPA binds to single-stranded DNA and stabilizes it. The replication fork contains helicase, polymerase, sliding clamp, clamp loader, and many regulatory proteins. This entire machinery coordinates accurate DNA replication. DNA polymerase in eukaryotes. Eukaryotic cells contain several DNA polymerases with specialized functions. DNA polymerase alpha is associated with primase and initiates replication. DNA polymerase delta mainly synthesize the lagging strand. DNA polymerase epsilon mainly synthesize the leading strand. DNA polymerase gamma replicates mitochondrial DNA. DNA polymerase alpha lacks proofreading activity. However, DNA polymerase delta and epsilon poses 3' to 5' exonuclease proofreading activity, which increases fidelity. An important concept is polymerase switching. Initially, Pol alpha begins synthesis, but elongation is later carried out by Pol delta and Pol epsilon. Primer synthesis and polymerase switching. In eukaryotes, primer synthesis is performed by the Pol alpha primase complex. Primase first synthesize a short RNA primer approximately 10 nucleotides long. Then, DNA polymerase alpha extends this primer with a short DNA segment. Therefore, eukaryotic primers are hybrid primers containing RNA followed by DNA. After primer synthesis, PCNA recruits the elongation polymerase. Pol epsilon synthesize the leading strand continuously. Pol delta synthesize the lagging strand discontinuously. This replacement of Pol alpha by Pol delta or Pol epsilon is called polymerase switching. PCNA and RFC. PCNA stands for proliferating cell nuclear antigen. It functions as the sliding clamp in eukaryotic replication. PCNA forms a ring-shaped structure around DNA and keeps DNA polymerase attached to DNA. This dramatically increases processivity. Replication factor C or RFC functions as the clamp loader. RFC uses ATP hydrolysis to open the PCNA ring and load it onto DNA. After loading, RFC dissociates and PCNA remains bound to DNA. A comparison is important. Bacteria use the beta clamp, whereas eukaryotes use PCNA. Leading and lagging strand synthesis. DNA polymerase synthesize DNA only in the 5' to 3' direction. Because the parental strands are antiparallel, two different synthesis modes occur. The leading strand is synthesized continuously toward the replication fork. This strand is mainly synthesized by DNA polymerase epsilon. The lagging strand is synthesized discontinuously away from the replication fork. It forms Okazaki fragments synthesized mainly by DNA polymerase delta. Eukaryotic Okazaki fragments are short, approximately 100 to 200 nucleotides long. Their short length is associated with nucleosome organization, primer removal and flap model. After lagging strand synthesis, RNA primers must be removed and replaced with DNA. Unlike bacteria, eukaryotes do not use DNA polymerase I. Instead, RNase H removes most of the RNA primer. DNA polymerase delta then displaces the remaining RNA region, generating a single-stranded flap structure. This flap is recognized and cleaved by flap endonuclease 1 or FEN1. After removal, DNA ligase seals the nick by forming a phosphodiester bond. This mechanism is called the flap model of primer removal. Primer maturation is essential for producing an intact lagging strand. Fidelity and proofreading. DNA replication must be extremely accurate to preserve genetic information. Replication fidelity is maintained through several mechanisms. First, DNA polymerase preferentially select correctly base-paired nucleotides. Second, DNA polymerase delta and epsilon poses 3' to 5' exonuclease proofreading activity. If an incorrect nucleotide is inserted, the polymerase pauses, the incorrect base is removed, and synthesis resumes. Third, mismatch repair systems correct errors escaping proofreading. Together, these mechanisms reduce the mutation rate dramatically. DNA polymerase alpha lacks proofreading ability and therefore functions mainly during initiation. Telomere replication problem. One major challenge in eukaryotic replication is replication of chromosome ends. Because chromosomes are linear, after removal of the terminal RNA primer from the lagging strand, DNA polymerase cannot fill the resulting gap. This leads to progressive chromosome shortening after each cell division. This problem is called the end replication problem. To solve this issue, eukaryotic chromosomes contain telomeres. Telomeres are repetitive DNA sequences located at chromosome ends. In humans, the repeat sequence is tag. Telomeres protect chromosome ends from degradation, fusion, and recombination. Telomerase and telomeric proteins. Telomerase is the enzyme responsible for extending telomeres. It is a ribonucleoprotein complex containing RNA and protein components. Telomerase acts as an RNA-dependent DNA polymerase or reverse transcriptase. Its catalytic subunit is TERT, telomerase reverse transcriptase. Telomerase carries its own RNA template and extends the G-rich strand of telomeres. After extension, normal replication machinery synthesize the complementary strand. Telomeric proteins form the shelterin complex. Important shelterin proteins include TRF1, TRF2, POT1, TIN2, TPP1, and RAP1. These proteins protect chromosome ends from being recognized as DNA damage. Most somatic cells show low telomerase activity, leading to gradual telomere shortening and aging. In contrast, many cancer cells reactivate telomerase, enabling unlimited proliferation. D-loop replication in mitochondria. Mitochondrial DNA replication occurs through specialized mechanism called D-loop replication. Mitochondrial DNA is circular and contains heavy strand or H strand and the light strand or L strand. Replication begins at the D-loop region. The H strand is synthesized first. When approximately 2/3 of H strand synthesis is complete, L strand synthesis begins from separate origin. Replication is generally unidirectional. Important enzymes involved include DNA polymerase gamma and twinkle helicase. The D-loop region has a triple-stranded structure and plays important roles in replication and transcription regulation. Finally, let us revise the most important high-yield concepts. Eukaryotic chromosomes are multirepliconic. ORC recognizes replication origins. MCM2/7 acts as the helicase. Pol-alpha synthesize RNA-DNA hybrid primers. Pol-epsilon synthesize the leading strand. Pol-delta synthesize the lagging strand. PCNA acts as the sliding clamp and RFC acts as the clamp loader. RNase H and Fen1 remove RNA primers. Telomerase is an RNA-dependent DNA polymerase. TERT is the catalytic subunit of telomerase. Shelterin protects telomeres. Mitochondrial DNA replicates through D-loop replication. Thank you. If you found this video helpful, please like, subscribe and share it with your friends.